Hi all, welcome back to CSEC Biology with Mr. Charles. Today we are continuing our deliberation on transport and circulation in humans. Okay, we would start off by looking at transportation of carbon dioxide in blood. Okay, let me just get my pointer, my pen actually. So carbon dioxide diffuses from respiring cells or cells of the body where respiration is occurring. Remember, carbon dioxide is a waste product of respiration. So the carbon dioxide would diffuse from the tissues into the bloodstream, and that would occur through the capillary walls. Now, most of the carbon dioxide is carried in blood plasma. Okay, and they would be carried in the form of hydrogen carbonate ions. And these hydrogen carbonate ions are also called hydrogen bicarbonate ions. Okay, or bicarbonate ions, HCO3- bicarbonate ions. Okay, so hydrogen carbonate is also called bicarbonate. Now, a small amount of carbon dioxide is also transported in hemoglobin. Remember, hemoglobin is a red pigment in blood, in red blood cells particularly. So the blood containing carbon dioxide would return to the heart via the veins. Okay, in the systemic circulation, it would return to the heart via or by means of the inferior and superior, superior and inferior vena cavae. Okay, and in the pulmonary circulation, the blood would return to the left side of the heart via the pulmonary veins. Okay, so the vena cavae would carry the blood containing carbon dioxide to the right side of the heart. Okay, and blood containing oxygen would return to the heart from the pulmonary circulation. Okay, that's the left side of the heart. Okay, so the blood containing carbon dioxide, when it returns to the left side of the heart, to the right side of the heart actually, it is then carried to the lungs for oxygenation via the pulmonary artery. The pulmonary artery would take it to the lungs, it would become oxygenated, the carbon dioxide would diffuse out of the blood into the alveoli of the lungs, and then the oxygen in turn would diffuse into the blood from the alveoli. The blood would become oxygenated, the carbon dioxide would be exhaled, and the oxygenated blood would be taken back to the left side of the heart, specifically the left atrium, where it can then be carried to the left ventricle and then pumped through the systemic circulation to the rest of the body. Okay, that's oxygenated blood. So it returns to the heart via the pulmonary vein. Good. Let us look at the functions of white blood cells. Remember, the white blood cells we're looking at are the broad categories. We have lymphocytes and phagocytes. So we want to know their role in the immune system. Now, before we go into the lymphocytes and phagocytes, I must introduce you to the term antigens. Now, antigens are cell surface markers. Cell surface markers, okay, they are found on the surfaces of cells. So let's say you have a cell here. This cell has an antigen on its surface. Okay, we have another cell here. This cell also has an antigen on its surface. Let's say this one here have another antigen here. Okay, an antigen here. And let's say this is another of the same type of cell. Okay, and we have another cell here also. We have an antigen here. We have an antigen here. We have one here. Okay, so these are foreign cells. Now these foreign cells have foreign antigens on the surface that the body's immune system recognize as being invasive or invading. So what would happen is that after they are recognized as foreign, the antigens are recognized as foreign, the B lymphocytes, okay, the B lymphocytes would secrete or produce antibodies that is able to begin the process of destruction. Okay, the antibodies would bind to the antigens. Okay, so you would have an antibody. Let's say this antibody here binding to this antigen. The antibodies are Y-shaped. You have another antibody here, okay, binding to this antigen. You have another antibody here, okay, binding to this antigen, okay. You have another one binding to this one. So the antibodies would bind to the antigens, okay, one binding here, okay, and so forth. You would have an antibody binding here, okay, you would have one binding here, okay, and then after the antibodies bind with the antigen, the antibody antigen complex would be formed. So we have an antibody antigen complex here. We have another antibody antigen complex here. One here. Okay. This is an antibody antigen complex here. Here also. So all these antibody antigen complexes would bind, okay, the antibodies that form the antibody antigen complexes would bind with other antibodies. So if you look carefully at the illustration I've just drawn, you would notice that the tail of the Y's are connected. So let me get a different color ink here. I'll use black. So you see, look here, we have two antibodies binding to each other here. You have three antibodies binding to each other here. So you get a clump forming. So this whole thing here is called a clump. Okay? And the process of forming the clump is called agglutination. Okay? The process of forming the clump is called agglutination. And I'll just write that in a different color. Okay, the process of forming the clump is called agglutination. The process, okay, see, of clump formation is called agglutination, right? So we say that the antibody antigen complexes agglutinate to form a clump, and that clump, so let's say this is a clump here, okay, let's say we have, well, let's say this is a clump here. We'll use this one, a macrophage, okay, which is an example of a phagocyte, would now come and it would surround the clump like this. Okay, it would surround the clump. Okay, it would surround the clump, and when it surrounds the clump, okay, after a while that clump would become completely engulfed. Okay, so you would have you would have the macrophage. Okay, you would have the macrophage closing up around the clump, and then inside here you would have the clump on the inside, and then you would have the what you call it, phagosome. Then here you would have a few vacuoles that would secrete enzymes that would digest the phagosome on the inside. Okay, and that is how the destruction of the foreign invading element, example viruses, are destroyed. Okay, so you would have the secretion of, okay, these are vacuoles, vacuoles, vacuoles. Okay, these are vacuoles. This is the phagosome. Okay, phagosome. Okay, phagosome, S-O-M-E. You have the phagosome, and then here you have the phagocyte. Okay, so the phagocyte engulfs the clump, or it swallows a clump, forming a phagosome on the inside. The phagosome is then digested by the phagocyte, and this happens because the vacuoles inside of the phagocyte would secrete enzymes. Okay, would secrete enzymes. So these would secrete enzymes that would destroy the phagosome. Okay, they would secrete enzymes destroying the phagosome. Okay, so the vacuoles, okay, secretes enzyme E. Okay, so the vacuoles secrete enzymes to destroy the phagosome. Okay, and that is how white blood cells functions. Okay, so the lymphocytes produce the antibodies that bind antigens. The antigens clump together, okay, in a process called agglutination, right? Agglutination, and the clump is then engulfed or swallowed by the phagocytes. Okay, example macrophages. The phagocytes then secrete enzymes from the vacuole that destroy the phagosome that is formed on the inside, right after the phagocyte swallows or engulfs the virus. Okay, and that is how antigens, that is how white blood cells function. Okay, good. Let us look at the structure-function relationship of different blood cells. It's basically a revision of what we have explained before in this video and in previous videos on the same topic. Okay, now red blood cells, they are small. Okay, they shape like a biconcave disc. They have no nucleus and no mitochondria, and the cytoplasm is filled with hemoglobin. Okay, these structural properties are important because it means that they can change their shape and fit easily through capillaries. They would also have more space to fill with hemoglobin so that they can transport as much oxygen and carbon dioxide as possible, mainly oxygen and a few carbon dioxide. The phagocytes, however, they are large. Okay, the nucleus is lobed, so they have a lobed nucleus, multiple lobes of the nucleus. Okay, and that is specifically important so that they would be able to, okay, leave the blood through small gaps in capillary walls. Okay, remember in one of the earlier videos we stated that capillaries have tiny holes in the walls that allow materials to pass or to be exchanged between the blood and tissues / cells. Phagocytes, the cytoplasm, okay, they do have mitochondria in the cytoplasm, and they have many small vacuoles containing enzymes. Okay, enzymes. Remember, the red blood cells have no mitochondria and they have no nucleus. The phagocyte, on the other hand, they do have a nucleus, although it is lobed. Okay, they do have mitochondria. Okay, so if you are asked to differentiate between red blood cells and phagocytes, then you know that's a key difference. Red blood cells are also small, white blood cells are large. Okay, now the vacuoles contain enzymes to digest bacteria, okay, and some viruses. Okay, and if we go back to the previous slide, you would notice that in this diagram here, this is the phagosome of the phagocyte, right? That's formed when the phagocyte engulfs the bacteria. Then we have the vacuoles on the inside that secretes enzymes to digest the phagosome. Okay, and that is how the bacteria / virus or virus is destroyed inside the phagocyte. Okay, now we also have lymphocytes. They are small cells, okay, and they have a small quantity of cytoplasm with few mitochondria. Okay, few mitochondria. However, when they are activated during an infection, although the cytoplasm is large, although the cytoplasm is small, it would get larger. Okay, so the cytoplasm enlarges during an infection so that you'll be able to produce more antibodies. Okay, more antibodies. And these antibodies are made of proteins. Okay, that's why your lymph nodes get swollen during an infection. It's because your lymphocytes, the cytoplasm, get larger, so you'll be able to produce more antibodies to fight against the infection. So that is why your lymph nodes, okay, get larger during an infection, because your lymph nodes is concentrated with lymphocytes. Okay, good. The next blood cells we will look at is blood platelets. They are the smallest of all the blood cells. Okay, so these purple cells here, these are platelets. Notice the spikes in the membrane. Okay, now blood platelets are important in blood clot formation. Right, this should be CLOT. Take note of that, blood clot formation. Okay. Okay, blood platelets, as identified before in a previous video, they are also called thrombocytes, and they are responsible for blood clot formation, as I stated a while ago. Now, thrombo, right, the root word thrombo that forms the term thrombocytes means to clot or clot. Okay, so that's just to let you know how appropriate the name thrombocyte is for blood platelets. Now, if you have a low blood platelet count, that would eventually result in prolonged and uncontrolled bleeding whenever you have a cut in your blood vessel. Okay, so with all blood platelets, your blood would not be able to clot at the site of an injury, and you would most likely bleed to death. Okay, if interventions are not taken as quickly as possible. Okay, you run the risk of bleeding to death. Now, let's look at the mechanism involved in the clotting of blood. Now, at the site of an injury, okay, at the site of an injury or an injured or broken blood vessel, thromboplastin is released. Now, this thromboplastin is an enzyme that converts a precursor molecule or a precursor molecule prothrombin, which is inactivated. It would convert it into thrombin. So thromboplastin converts prothrombin into thrombin, and this thrombin in turn converts inactive fibrinogen into fibrin. And fibrin forms the mesh that traps blood cells around the site of an injured blood vessel, forming a plug that blocks the blood vessels. Alright, so that is how the clotting of blood occurs as a mechanism of action. All this happens very quickly, in less than no time. So again, thromboplastin converts prothrombin into thrombin. Thrombin then converts fibrinogen, which is inactive, into fibrin. Notice here it says inactive fibrinogen into fibrin. So make sure you study this slide very carefully. This is a very important slide. And actually that was the last slide. Thank you very much for paying attention to this recording. It was a pleasure. Do enjoy.